Okay. So now we have spoken about the architecture and some differences in the light path already. And now we're going to talk about what are the differences between wide field and single point confocals.
Sylvie,
Yes?
we are again seeing the presenter view.
Oh, sorry. Let me see. How do we do that? So here I go escape. And I'm sharing with the wrong stuff. Too many screens. Let me see. Did that one then. Let's check. Is that okay? Yeah? No, you
Yes.
see? Okay, thank you. All right. So yes, we're going to go about wide field and single point confocal. So we're moving through our flow. So I remind you that this is the microscope stand. It's called actually microscope body as well sometimes. And the rest is the equipment. And we have hard connections. And this is what we have to recognize. So the equipment that is attached by hard connections to the microscope body. And so they can be cameras or they can be scanners. And there's a few more, but pretty much is cameras and scanners. So we're going to have a look at that. And we have seen that we have microscopes without camera. And if they can do imaging, they are single point confocals. And the microscope with a camera are all other types of microscopes. And if they do not have a scanner, they are wide field. And if they have a scanner, they are multi-point confocals, also called spinning disk confocals. And so there's a few more, but more or less that's about it. So all microscopes, all modern microscopes acquire digital images. And so we are going to have a tiny introduction, let's say, to digital images now so that we can understand how we acquire our images. So what is a digital image? We have a digital image is a matrix of picture elements. This is why what pixels come from. It's picture elements. So the little blocks that make a picture are pixels. Each pixel has an XY coordinate and one intensity. So the intensity is divided in steps. So we have only a finite number of possible values. And the number, so it starts at 0 and all the way to a certain number, which is defined by the bit depth. So we will speak about bit depth next week. But if you acquire images in 8 bits, then we have 256 integer values. And so it goes from 0 to 255. So we have altogether 256 values. And you cannot have an intensity value of 200.3, for example. It's always integer values. Okay, so we have a finite numbers of possible values. So if we zoom into this area in the image, depending on how the image is acquired, we could see small pixels like this or big pixels like this. So the way we acquire the image also has an influence on the image. And we will also talk about this next week. So if we look a little bit at these pixels, we have, so each pixel is, so a picture element is one of these little squares, and they have a single X coordinate, a single Y coordinate, and a uniform intensity. So each of these pixels has one intensity. And so then we have this matrix of pixels, each of them with an X, a Y, and an intensity. So that means that we need to have a device that records the XY coordinate, and we also have to have a device that records the intensity. Sometimes it's the same device that does both, and sometimes it is not. So in each image, we have, each image contains 2 different types of information. It contains a matrix of millions of pixels, which has each an XY and an intensity, but it also contains metadata. And metadata is information about the way you acquire your images. It can be, for example, which filter you had there so that you can see which color you were actually imaging, or at which Z level your microscope was when you acquired this particular image. It can be all the settings that you had on your microscope. So the metadata is embedded inside the image. And it is very important. This is the reason why I've been insisting a lot when you submitted your images. I want the original, the images in the original format that we use, that is recommended by the manufacturer of the microscope, because this format definitely preserves the metadata. So if you just randomly choose JPEG or TIFF, for JPEG, you will definitely lose the metadata, but for TIFF, you may also lose the metadata, or it's scrambled. It also happens quite a lot. So it's really important to save the images in the original format so that you do not lose the metadata. And I think you have already checked, and I think some of you are missing the metadata. And so this is something we have to solve together. Okay, so microscopes with camera and without camera. So the sensor, if we do not have a camera, in this course, we're going to call the sensor a detector. So I remind you that in this course, we're going to call sensor anything that can capture light, for example, our eyes, detectors, or cameras, okay? So in a microscope without camera, the sensors are detectors, and they only acquire 1 pixel at a time. I remind you that the microscope without a camera and that can do imaging is a single point confocal. So we have single point imaging. So 1 pixel at a time. Every time we image is only 1 pixel. And then we move to the next pixel and we image it. The detectors always record black and white images. So we play with the filters and the dichroic mirrors, right, as we have seen yesterday. And one single point confocal can have up to 34 detectors per microscope. So each of these detectors that record 1 pixel, but we can have up to 34 of them. But if we have 34 detectors, each of them records 1 pixel. So they will record different colors for 1 pixel, but it's always 1 pixel that is imaged at the same time, even when we have 34 detectors. So we will see all this in the workshops that are coming and the lectures that are coming, how this works. And there's only one type of microscope with detectors. They are single point confocals. The detectors on single point confocals only record the light intensity. So we said each pixel has an XY coordinate and a light intensity, and the detector only records the light intensity. So we have 1 pixel and only intensity. And so we always require a point scanner which records the XY coordinate. Where is this pixel amongst the whole image? This is the job of the scanner. The detector gets the intensity and the scanner gets the coordinates. This is the type of names that these detectors can have. So on your microscope, you may recognize some of them, like high-D detectors, HPD detectors, PMT, GASP, etc. All these are detectors that you can find on a point confocal. Now, if we look at the microscopes that have a camera, the sensor is a camera. Each camera records several millions of pixels at the same time. And that's, so several millions versus one, right? I didn't put several millions in here, but it is. So the cameras can be either black and white or color, but the color camera are less sensitive than the black and white camera. So for high-end applications, that what we do in general, we would use a black and white camera. And then at the hospital, very often they use color cameras. We could have up to 4 cameras per microscope. It's quite rare, but it does happen. 2 cameras even is rare, but it does happen as well. On one of our systems, we have 2 cameras. And in that case, it's 4 images of millions of pixels, okay? But it could be that the different cameras record different colors. So we could have like a dichroic mirror that sends the green light to one camera and the red light to another camera, and so you can image them at the same time. But most of the time, we only have 1 camera per system. And the microscopes with camera is all the other types of microscopes compared to the single point confocal. So all the other types of microscope work with cameras, wide field systems, multi-point confocals, the spinning disk confocals, or light cheese microscopes all work with cameras. The camera records both the light intensity and the XY coordinate. So this is the whole image. this is only part of the image. So this is a small bit of the image, and here will be the whole image. So each of these has an XY coordinate and an intensity, and this is the job of the camera to record them. So that's what we have here. And these microscopes with camera, they can either have a scanner or not. So on the single point confocal, we always have a scanner, which is the one that records the XY coordinate. But on the microscope with a camera, sometimes we have a scanner, sometimes not. And this is the type of names that you may recognize when you look it up. You look up your camera online. They could be called CCD or SCMOS or EMCCD, and sometimes we have front illuminated all this or back illuminated all this. So you can have a front illuminated SCMOS or back illuminated SCMOS. So these are different types of cameras, and when you will look up your microscope online, you may find this type of information. So I want you to just have seen it so that you can recognize it. So basically, your microscope will be mostly single point confocals or wide field. And we also have some light sheet systems. And so the tricks that help both type of confocals on one hand, so this one should come up a little bit later, but I forgot my animations, it's okay. So the speed, the camera system is much faster than a single point confocal. So there are some tricks that can make a single point confocal go faster. For example, we can image bidirectionally. A lot of people who image on single point confocal are not aware of that. We could image with a resonance scanner if we have one on our system, but also we have several detectors on a point confocal, and that can help us image several colors at the same time and therefore go faster. And then we can do also spectral mixing. Another difference between single point confocal and camera-based systems is the flexibility. Single point confocals have a very high flexibility. You can choose the pixel size, you can choose the pinhole size and the scan area and even the scan shape, just the way you want. You don't have that flexibility on a camera-based system, but there are some tricks to add a bit of flexibility when imaging with a camera. And that can be to change the size of the field of view. You could crop the camera. That's one possibility. And then you have also flexibility of the pixel size. Sometimes on some microscopes, you have an extra magnification lens that can be added to change the pixel size, or you can have also binning. You can bin the camera. So we will talk about all this later so that you know what it is. But these are ways to improve the flexibility on camera-based systems. The pinhole size, if we have a spinning disc, it also has pinholes because it's a confocal. And sometimes you can get a system with different pinholes, different discs, let's say. Maybe you have 2 discs, so you have a little flexibility there. And the flexibility of the emission filters, we have a great flexibility on point confocals, but sometimes on camera-based systems, we could have several cameras, as I said, to image several fluorophores at the same time, or you can even split the camera chip in 4 and image one color on each of them. So we can add a little bit of flexibility to camera-based systems, but mostly single point confocals are much more flexible. And we will talk about all this later in these workshops. So the conclusion of this is that if you use a single point confocal without using the flexibility, so without adjusting all the flexible parameters like the field of view, the pixel size, the pinhole size, and the filters, you would get a lot in using a multi-point confocal. So a lot of people use single point confocals because they think this is the best microscope, but because they do not know how to set the settings, they use it in a very under-optimal way, and they would be much better off using a camera-based system in that case. So my aim is to make you realize that either you should go to a camera-based system or you should use your single point confocal in a correct way. Okay, and now this is a little for people who have never imaged a full field of view with lots of pixels on a single point confocal, I thought that they should experience how slow this can be. So this is a full field of view with 4,000 by 4,000 pixels, and it takes 23 seconds to take one image. And bling, this is what we get when we image with a camera. It's instantaneous, and we can have also all these pixels. So I think that there is a lot to be said for camera-based systems. And again, if you do not use the flexibility of a single point confocal, consider swapping to a camera-based system and your life will be easier. So let's have a look now at microscopes with cameras. So we have wide fields. How does a wide field system work? So basically wide field systems, so microscope with cameras, it's all microscopes except the single point confocal. Wide field is one type of microscope with camera. So here we have our sample and we have an objective. And the objective is here. Is this an upright or an inverted system?
Inverted.
Inverted, because?
The objective is under the sample.
The objective is pointing upwards, and so it's under the sample, and therefore it is an inverted system. Ta-da! So we have here, the objective is in focus. So we have a focal plane for the objective, a specific plane where it is in focus. Now we have no light, so the sample is completely black. And we have a lamp here. So it could be a mercury lamp. It's called also metal halide lamp. It's also a type of mercury lamp or an LED. But we have a light source, and then it sends some white light and it reaches the excitation filter, so more or less white, it depends on the LED, but anyway, LEDs also have excitation filters, and which filters the light, and that goes through the objective and illuminates the sample. And you could also have a laser. And in that case, you do not need the excitation filter, right? This is what we say because lasers are monochromatic. So we go back to our little lamp with our excitation filter, and the light reaches the dichroic mirror. And in this particular case, we have a dichroic mirror that transmits the excitation light. So as I said, we can have two ways. And so we remember that we have the three roles of the dichroic mirror. It sends the first one is that it sends the excitation light to the sample. So this is what it does now. And then, of course, some of the light is emitted, and so it will go towards the sensor, and the reflected excitation light is going to get lost in the microscope, so it doesn't go to the sensor. So our sample gets excited, and what happens is that the whole sample gets excited at the same time. This is because we have a wide illumination. We do not have a concentrated illumination like in the single point confocals. We have a wide illumination, so the whole sample is illuminated at the same time. So this is why it's called wide field. So if we look in here at this image, we can see here we have a fluorescence solution, and this is the source of the images, by the way. So we have the fluorescence solution here and the objective. This is an upright system because the objective is pointing downwards, so it's above the stage. And here we have a little fish. And what we can see is that we're shining light on this fluorescent excitation light on this fluorescence solution. And here we see the shape of the illumination. So the illumination is wide. It's a wide field of illumination, and the whole sample here is going to be excited. And it's excited both in many places in X, Y, but also in Z. So the sample is illuminated everywhere, okay? So the excitation concerns all the XY in the field of view, plus all the Z as well. So our sample gets excited and it starts fluorescing. And so we get some light that comes back and it reaches the dichroic mirror, which sends this light towards the emission filter. And then the light continues. So because we have we have a wide excitation, we have a defocused excitation, we also have a defocused emission. So we don't have just one point of emission, it's like all the light comes in at the same time. And this is projected on the camera chip, which records the light wherever it comes from. So this camera chip will record both the XY coordinates of the light and also the intensity for each pixel. And this is the type of image that we get. In general, cameras, especially nowadays, they have a lot of pixels. So 2048 by 2048 is actually now little. You can get much larger chips than that. And that allows you to acquire lots of pixels at the same time. And this is like instantaneous. So you get this image more or less. And now, so imaging with a camera is a little bit like flash photography. So you're going to take a picture of your friends with your phone, you have a flash, and boom, suddenly all the light goes and everybody is illuminated at the same time, and the light comes back to your camera. So it's instantaneous. We get all the pixels at the same time. And so this is, if we look a little bit closer at this image, we have, it's made of pixels, and we look at it, each of them has an XY coordinate and an intensity, and this is what the camera records. Okay, but we can see that all the Z levels are excited. So the plane of interest might be here. The plane that we're imaging is here. This is the focal plane, okay? But actually the sample above and below is also excited, and therefore it also fluoresces. And that creates a haze in the image, and you can see that. And if we compare with a confocal image where we have so it can be a single point confocal or multi-point confocal, a confocal image where we have an optical section. This is what the image looks like, okay? So we're going to be comparing. This is what a wide field with a slightly thick sample would look like, and this is if we have an optical section. So we will talk about optical section later. Now let's have a look at the microscope with detector. So now we're talking single point confocal. So the principles that I've shown you in this one are the same for the other systems as well. So the microscopes with camera, but this was in wide field, and then we will see later how the spinning disk and the light sheet work. But the principles are the same. Here we have detectors. So now it's a very different system. So we have our sample, we have our inverted system with the objective pointing upwards. The sample is not illuminated now, so everything is black. And we have lasers. So the lasers come in and the lasers come in and they shine some light. They reach the scanner. And inside the scanner, there is a dichroic mirror. And this dichroic mirror is going to send one of the laser light through the objective, because the excitation always goes through the objective, right? And it goes to the sample. So the sample gets illuminated, only one small spot. And so this is a single point of excitation. And if we look at it again with this very clever way with this chamber filled with fluorescent dye, we can see that the shape of the illumination is very different. Now we have like a double inverted cone. Okay, so we have these cones of light, and it's focused here. Yes, it's focused here. This is the highest focus of light. So this is where the light is most concentrated and strongest. But we anyway illuminate above and below. So because we illuminate above and below, these areas are also that are out of focus will also fluoresce and will send their emitted light as well. So now we have the excitation is a small XY point only, and the shape of illumination is a double cone in Z. And this is to be compared with the wide field illumination, where we have all XY, so not a spot, but all XY and all Z illuminated at the same time. Right, so now our sample starts fluorescing, and it sends from this little spot that is excited, it sends the emitted light again through the objective. So both the excitation and the emission light go through the objective, and the light reaches the scanner again with the dichroic mirror. And so the scanner is going to send this emission light to a spot through the filter, the emission filter. Or if you have a spectral detector, you can have up to a number of detectors, let's say minimum 2 or 34. And if you use spectral detection, you may not have an emission filter there, but this is sort of a common case as well. So we have the emission filter, and then we have all the detectors that we have. And we will see all this in the workshop and lecture called Imaging Multiple Colors at Once. We will see how this works. And then the image is slowly built up, one point at a time and one line at a time. So we have, so if you wonder, so we are building up the image slowly. Now, why is this called confocal? And what is a confocal? So we have a certain plane of focus for the objective. So this is the focal plane of the objective. And then we also have, so we have focused excitation, and we also have focused emission, because we have only one point that is excited, there's only one point that fluoresces. So it's a focused emission as well. And what we do is we put a big metal plate in the light path to block all the emission. Well, not all, but most of it. So we block the emission. We have just a little spot in the middle, little hole in the middle that will only allow a little bit of the light to go through. So it sounds pretty freaky, but there is a reason for that, clearly. But what I would like to tell you is that because we have here the focal plane for a pinhole, and we have a focal plane for the objective, and these two planes are actually linked to each other, so they are conjugated. We have two conjugated focal planes, and this is the reason why confocal microscopes are called confocal. So as long as you have the objective focal plane and the pinhole focal plane conjugated, you have a confocal. And on a spinning disk microscope, this is what happens. You also have pinholes, and you have a focal plane, and the planes are conjugated. So even a spinning disk is a real confocal. So if you have no pinhole on a microscope, it's not a confocal. Okay, so the excitation plane and the pinhole planes are conjugated. So what is the role of this pinhole? It reduces the out of focus light, and we will see a little bit how that works later. And this is what makes it very interesting for a confocal. So that's an important thing. So once we have had our, the first spot here is as fluoresced, this is what the scanner is sending to the detectors. The scanner is going to record the position, X and Y, and the detector records the intensity. And then, so this is what the detector records the intensity. And we get a little extra dot in our image. So we have an extra pixel that has the XY coordinate that the scanner has recorded and the intensity that the detectors have recorded. Then we move, the scanner moves the spot to a certain distance. So we jump from here to here. This distance is actually the pixel size. So we can move more or less. And this is the reason why in single-point confocal, we have the flexibility of the pixel size, because we can decide how much we move between spots. So now we're exciting this spot, and it's exactly the same thing. It starts fluorescing, and the scanner is going to record the XY coordinate of the second spot, and it will send the emitted light through the pinhole to the filter, the emission filter, if you have one. And there we go, the detectors record the intensity. And so all the detectors will record the intensity of this very same spot with the same coordinate, but each detector may record different wavelengths. Remember, lambda is wavelength. So we will have, if we have 34 detectors, we may have 34 wavelengths just for one single spot. And there we start building the image, and we put an extra one, and we keep it going like this. We get a third spot, we move again, and then we get another set of intensities and etc. And then we build the image like this slowly. And this is the reason why it goes slow, because we're building the image point by point. So the scanner scans the excitation light on the sample, and then the scanner also scans again the emitted light to send it to the detectors. So now that we understand a little bit better this slow building of the point confocal image, how do pinholes reject the out of focus light? So when we get the emission out, so the pinhole is in the emitted light path, basically, it's just before the detectors. And we get these spots that fluoresces will fluoresce in all directions, and only some of the light is going to go through the objective. As you remember, what happens inside the objective is that we have diffraction on the edges of the objective, and through all the lenses inside, all the way to the final image plane, where we have a very complex 3D interference pattern, which looks like this in the XY direction and looks like this in the Z direction if we image only a very tiny spot, this is what it looks like. So every single fluorophore in our sample is going to make an image like this. And this image is very predictable because light is predictable, the behavior of light is predictable, and this image is called a point spread function, okay? So we get a tiny fluorophore, and through our microscope, we're going to get a certain shape called the point spread function. And I've told you that if we draw a line in here and we look at the profile, this is what it looks like. So the point spread function is often represented this way, where we have like the little waves that we see here. And then the very bright center is here. But we need to remember that this is a 3D shape. So it goes in 3 dimensions. So it's like mirrored below. So here we have our point spread function. And what we do when we put the pinhole here is this. We hide all the rings, and we keep only the bright center. And this is what happens here. And if you wonder, when you set your pinhole size to 1 AU, this is what it means. 1 AU is 1 Airy unit. And 1 Airy unit, it depends on the wavelength. So the real size of this depends on the wavelength, but basically it's always at the bottom of this bright peak. We want to get rid of the rings, okay? And so this is what we do. We actually remove everything that is, all the rings that are around the spot. And this is what it looks like in the Z dimension. You remember my little rugby man? We still see it. And so now in the Z dimension, this is what we do. We only keep this bright ring. And so what happens for fluorophores that are out of focus or a little bit outside, with most of them, they will send only the rings that are all around the bright lobe. And so because we are filtering out, we only keep the things that the main lobes that are in the focal plane, and we throw away all the rest of the light, then most of this light is going to be eliminated. This is quite dim, so it will go through the pinhole, but this main lobe is going to be eliminated by the pinhole. So this is very useful to increase the contrast basically in the images. So you see, I toggle it a little bit so you can see what happens. For it's in focus and out of focus, we eliminate basically the bright lobe. This is the way the pinholes reject the out of focus light. So the out of focus light is greatly reduced, and the light from the in focus fluorophores is sent to the detectors. So this light is sent to the detectors only. Right, so now I've told you a little bit how wide field works and how single point confocal work and how we build the image slowly and how the pinhole rejects the out of focus light. And this is why we get an image that is sharper. So when we get the image from a single point confocal, we have to realize that the rest of the sample is illuminated. We have a double cone of excitation, double inverted cone. So we do excite the above and below the plane, but we throw away the light that is emitted from these planes using the pinhole, okay? We do not just excite the plane of interest. We actually excite also outside the plane of interest, we throw away the light. So here are the similarities between wide field and confocals. People have a tendency to think that the wide field system is very simple and maybe only used for live samples and only for transmitted light, but it's not true. So we can have upright or inverted microscope for both of them. We can use transmitted light imaging. We can use fluorescence imaging on both of them. And we can have, both systems can be fully automated in exactly the same way. They can do Z-stacks as well. And we have high resolution in XY. So this is something to remember, and we will talk about the resolution later, but we have a high resolution in XY also on the wide field system. And we can use fixed or live sample on both of them. And we will also talk a little bit why maybe it's not a good idea to use a live sample on a single point confocal, or let's say it would not be your first choice. And in the whole volume is bleached in wide field. So we have this big like cone, this big like cylinder of excitation that shines on the sample, and therefore even the planes above and below the plane of interest are bleached. They are excited, they emit light, and they are bleached. And in single point confocal, it's the same thing. The shape of the illumination is a little bit different because we have this double inverted cone, but we anyway bleach above and below the plane of interest. And we can have super resolution on both types of systems. So we can use SIM and turf and storm and palm on a system that is based on a wide field with sort of extra equipment. And we can also use super resolution on a single point confocal if we have the equipment to do STED microscopy. And we have someone in the course who does STED microscopy. So what are the differences now between the wide field and single point confocal? So wide field uses a camera, and therefore when it images, it acquires millions of pixels, and a single point confocal acquires 1 pixel at a time. So if we have several detectors, and we always have several detectors on a single point confocal minimum 2, if we have several detectors anyway, they all image the same pixels, but maybe different wavelengths. Wide field is very much like a snapshot, like flash photography. So it's instantaneous. Whereas in single point confocal, we build the image very slowly, point by point and line by line. Wide field has a wide excitation light, whereas the single point confocal, in single point confocal, the light is very highly focused on the plane of interest. So we have these double inverted cones, and the highest focus and the brightest light, excitation light, is at the plane of interest, but there is also quite a lot of light on the other planes. Wide field is very fast, and single point is usually very slow, but we'll see that we have some tricks there. And wide field, on the wide field system, usually, if you have only one camera, usually if you have multiple colors, you image them one by one. So you have to image sequentially. You first image the green and then you image the blue or something like this. But in a single point confocal, because we have several detectors, in general, we can combine colors and image them at the same time. So we have to be very careful and avoid bleed through. But here we need also to avoid bleed through. So in the wide field system, we have no pinhole. So these systems are extremely photon efficient. Even if we, like any photon that comes in can be captured by the camera, whereas in a single point confocal, there are a lot of photons that are emitted by the sample and that are lost because they are blocked by the pinhole. So we throw away quite a lot of light in a single point confocal. In a wide field system, all the Z levels are recorded at once. So we get this blur for the emission light that comes from the out of planes that are out of focus, then the emitted light becomes makes a blur in the image. Whereas in a single point confocal, we have an optical sectioning. So what is the optical sectioning? If you want to see only part of your sample, you can cut it physically. But you can also try to cut it with using optical tools. Like, for example, what we're doing with the pinhole, we are illuminating above and below the plane, but thanks to the pinhole, we are throwing away the emitted light that comes from the other planes. And therefore, it looks like we have cut our sample, but we still, without cutting the sample, it looks like we cut it. So it's an optical section. Sectioning the sample using optical tricks, let's say. In a wide field system, we have a low contrast if our Z is just a little bit fluorescent. So even if we have a monolayer of cell, we already see a difference between the same monolayer of cell image with a single point confocal compared to a wide field system, because the top, let's say if you focus at the bottom of the cells, the top of the cells anyway is going to fluoresce and create a little bit of blur. So as soon as we have a sample that is a little bit thick, even one cell layer, we will lose some contrast because of the blur created by the out of focus haze. Whereas in single point confocal, we get a very high contrast, even for samples that are thick. And so what happens is that we will talk about resolution, but basically you could use the same objective on both systems. And this means you would have the same optical resolution, okay, on both of them, because the optical resolution is dictated by the objective. So you get the same optical resolution, but because in a wide field system, you would have this blur, you will have less contrast. And so the usable resolution that we have on the wide field system is lower. So in Z, we have no usable resolution without deconvolution. So we have the optical resolution, but the actual resolution that we can use is different. Whereas in a single point confocal, we can acquire all the optical resolution that is delivered by the objective. So we have a high resolution in Z even without deconvolution. So that is one of the major differences between single point confocals and wide field. A wide field system is very fast imaging at high XY resolution, but the single point confocal is very flexible. So this is why I'm saying that this is one of the major advantages of a single point confocal. You have to use it, otherwise you shouldn't use a single point confocal. On a wide field system, we can have very high sensitivity, like single fluorophore sensitivity, single photon. Also, but in a single point confocal, we can do spectral imaging, which is not possible on wide field systems. So this is an advantage. So of course, a wide field system is simpler, so it's cheaper. And a single point confocal is more expensive, it's more complex, and it requires a little bit more tweaking, let's say. So mostly on the systems that we have here, we have either, the main differences are we have either camera or a few detectors, so millions of pixels or 1 pixel only. And this is like flash photography, whereas this is built point by point and line by line. And otherwise, we have no pinhole, so it's very photon efficient for wide field. And the pinholes, so we throw away the autofocus light on a multi-point confocal. And here, that means that we have a blurry image if the sample is just a tiny bit thick. And here we're going to get an optical sectioning. So there are other ways to remove the autofocus light. And so to get this optical sectioning effect, in multiphoton microscopy, we can do that. And multiphoton microscopes are based on single point scanning confocals. And also in spinning disk confocals, they are camera-based, but we also have an optical sectioning. And light sheet system, they are also camera-based, and they have also optical sectioning. So it's only the wide field systems that do not have optical sectioning capacities. All the others have it. Okay, so now I'm going to quickly look at my time sheet and see what's going on. And yeah, it's good. So I think we're going to go now to the other Zoom, and we're going to do some quizzes together. And then we will be back in this Zoom at 11:25.